Have you ever wondered why children do not look exactly like their parents, or why some siblings look so different from each other? The answer lies in the process of meiosis, which generates the gametes for reproduction. Meiosis is the process by which haploid cells are produced from a diploid cell. For this to occur, the chromosomes must be correctly sorted and distributed in a manner to create genetically unique cells with half the number of chromosomes as the original cell. Meiosis occurs in special cells called germ cells within the gonads of males and females. For the successful reduction in the number of chromosomes to occur in the new haploid daughter cells, 2 rounds of divisions are necessary. These divisions are termed meiosis I and meiosis II. Although the processes of mitosis and meiosis look similar, there are a few major differences. One obvious difference is that mitosis results in 2 diploid daughter cells, and meiosis results in 4 haploid cells. As in mitosis, meiosis begins after a cell has successfully completed the G1, S, and G2 stages of interphase. During the S phase of interphase, the DNA is replicated, producing 2 copies of each chromosome called sister chromatids. The paired centrioles in the cytoplasm duplicate and begin extending microtubules that will form the meiotic spindle. Sister chromatids remain attached at the centromere and condense as the cell enters prophase I of meiosis. Up to this point, the cell looks similar to mitosis, but 2 events occur in meiosis that do not occur in mitosis and lead to genetic diversity. The first event occurs during prophase I, when homologous pairs of sister chromatids lie side by side in a process called synapsis, forming a tetrad or bivalent. The homologous chromosomes share similar but not necessarily identical genes. Once this structure is formed, the second event, called crossing over, can occur. During crossing over, a physical exchange between chromosome segments of non-sister chromatids occurs, increasing genetic diversity. Prophase I concludes with the fragmentation of the nuclear envelope as the duplicated centriole pairs move to opposite poles of the cell. As they move, the centrioles extend spindle fibers forming the meiotic spindle. In prometaphase I, with the paired centrioles in place, the meiotic spindle is fully formed. The sister chromatids attach to the spindle fibers by their kinetochores. Here, another key difference between mitosis and meiosis occurs. Because of synapsis and crossing over in meiosis, homologous chromosomes remain aligned, so that a pair of sister chromatids is attached to only one pole by the kinetochore microtubules. In mitosis, each sister chromatid is attached to a spindle fiber. The fibers originate from opposite poles. During metaphase I, bivalents randomly align along the metaphase plate. Due to independent assortment, this alignment is random and adds to genetic diversity. In anaphase I, the homologous chromosomes separate and move toward opposite poles. Meiosis I ends with telophase I, when the chromosomes decondense and the nuclear envelope reforms. Cytokinesis separates the cytoplasmic material, and the 2 daughter cells are separated by a cleavage furrow. Since the final product of meiosis I is 2 haploid cells, meiosis II begins without the chromosomes going through another round of DNA replication. Centrioles again duplicate and begin moving to opposite poles of each cell. In prophase II, the sister chromatids condense while the spindle starts to form as the nuclear envelope disappears. In prometaphase II, the sister chromatids attach to the spindle by kinetochore microtubules, with sister chromatids attached to opposite poles. The spindle aligns the sister chromatids along the metaphase plate during metaphase II. During anaphase II, sister chromatids separate and individual chromosomes move toward the poles. The entire process ends with telophase II, as the chromosomes decondense and the nuclear envelope reforms. Cytokinesis occurs, and cleavage furrows separate the 2 daughter cells into 4 haploid daughter cells. The haploid daughter cells will specialize into gametes, either sperm or egg. These fuse in fertilization to form a zygote, which will grow into a child. The child receives half its chromosomes from its mother and half from its father. Because men and women produce millions of gametes, and the selection of gametes in fertilization is random, this contributes to genetic diversity. This explains why a child isn't identical to either parent. But why aren't siblings identical? The diversity comes from several sources. In prophase 1, non-sister chromatids can exchange DNA through crossing over, increasing the genetic diversity of individual chromatids. In addition to crossing over, in metaphase 1, the pairs of homologous chromosomes align independently along the metaphase plate and sort independently into the daughter cells. This process, called independent assortment, produces 4 genetically distinct haploid gametes. As the total number of chromosomes increases in an organism, the number of genetically distinct gametes increases by 2 to the nth power. This means an organism with n equals 3 can produce 8 unique gametes. For humans where n equals 23, there are 2 to the 23rd power unique gametes formed, or over 1 million different possible combinations. The combination of independent assortment, crossing over, and the random pairing of gametes during sexual reproduction increases genetic diversity and explains why a child will not look exactly like his or her parents or siblings.
So firstly, meiosis in Greek. Meiosis, this word meiosis is derived from Greek word meiosis, which means lessening or decreasing. So it is a process by which chromosomes in a cell nucleus is halved during the formation of gametes. So mostly ये जो division होता है, it occurs in the germ cells, और ये chromosome number को करता है half in the daughter cells. It forms the basis of sexual reproduction. जो भी organism sexual reproduction show करते हैं, उनके जो germ cells है, they will undergo this division, that is meiosis, and it also helps in increasing the genetic diversity of offspring. जो offspring होता है उसमें जो genetic diversity और variations हैं वो meiosis की वजह से ही होती हैं. Then genome is replicated once. इस केस में जो meiosis है, इसमें जो genome है that is replicated once and separated twice. और इसमें जो separation है 2 बार होती है और replication 1 बार होता है, that result in the formation of 4 cells. और एक meiosis के result से total 4 cells बनते हैं, that we'll discuss in upcoming slide in detail. So there were scientists like T.H. Morgan in 1911. He detected crossovers in meiosis. He was the first person to observe these crossovers in meiosis in Drosophila melanogaster. और इन्होंने linkage के ऊपर भी बहुत काम किया है, the concept of linkage. So he was the first person to observe crossovers in meiosis in Drosophila melanogaster. Then this is the photograph of T.H. Thomas Hunt Morgan. Next comes J.B. Farmer and J.E. Morey. In 1905, they derived the term meiosis. So they were the first person to coin the term meiosis. And if we want to study meiosis practically, so we use the floral buds. ये जो onion buds हैं, इनको हम study करते हैं to observe the meiosis under microscope. इसके लिए पूरा procedure follow करना पड़ता है, how we can look for these cells. जो buds हैं उनमें reduction होती है, meiosis होती है to form pollen grains, and they are undergoing this division, that is meiosis or meiotic division. So for studying meiosis, next comes types of meiosis. Basically 3 तरह की meiosis होती है. First is gametic meiosis. It occurs during the formation of gametes. For example, there is a diploid thallus. This is the mature body. Basically it is sporophytic. Plant body, it bears gametangia. जो gametangia है वो भी diploid होते हैं. जब इनमें gametes बनते हैं, during that time there is this division, that is meiosis, और 2 gametes बनते हैं. That may be isomorphic or heteromorphic, or these both the gametes are haploid. Then they will undergo the process of fertilization and will form the diploid zygote, which is also known as oospore, and it will grow into the mature diploid thallus. So this type of meiosis, which occurs during the formation of gametes, is known as gametic meiosis. It occurs in all multicellular animals and many protists. closely linked with the formation of the gametes. Next comes zygotic meiosis. In case of zygotic meiosis, the main plant body is haploid, that is gametophytic. Gametophyte होता है main plant body. It bears gametangia, which are also haploid. They will produce the gametes. One will act as the male gamete and other will act as the female gamete. Both of these gametes will fuse by the process of fertilization, will give rise to this diploid structure, which is zygote or oospore. Then this zygote will undergo meiosis and will form the haploid cell. They will grow into the haploid thallus. It mostly occurs in case of lower algae. Occurs only in protists and fungi. In some algae it also occurs. This division occurs just after fertilization. जैसे ही fertilization होता है उसके बाद ये meiosis होता है to produce haploid spores. This results in the formation of haploid spores. Next comes sporic meiosis. It occurs in some plants and algae. ये भी कुछ plants और कुछ algae में होता है present. This type of meiosis. It is related to either gamete formation or fertilization. So या तो ये gamete formation के साथ related है या फिर ये fertilization के साथ associated है. So there are basically three types of meiosis. That is gametic meiosis, then zygotic meiosis and sporic meiosis. In case of sporic meiosis, suppose this is the adult plant that is sporophyte. It will produce sporangia, and these sporangia will undergo the meiosis process and will give rise to this spore. This spore will germinate to produce haploid plant body, that is gametophyte. This gametophyte will bear the gametangias, which will produce male and female gametes respectively, and they will fuse by the process of fertilization and develops into the zygote. Zygote will give rise to this diploid generation, that is sporophyte. The best example is in case of funaria, that we can observe that the gametophyte will form the sporophyte, and it is partially parasitic in nature. और जो sporophyte है funaria का, that will bears the sporangia, means that will produces the spores after meiosis. So this is the next comes stages of meiosis. Meiosis is a very complicated process as compared to the mitosis. It includes two nuclear divisions. So it can consist of two nuclear division, and it is divided into two parts, that is meiosis 1 and meiosis 2. Meiosis 1 is further divided into its four substages, which include prophase 1, metaphase 1, anaphase 1, telophase 1. Out of these four stages, this prophase 1 is most complicated one, and it is further divided into 5 different stages, which include leptotin, zygotin, paketin, diplotin, and diakinesis. This is the slide which shows the animated pics of these stages of prophase 1 of meiosis 1. First is leptotin. Leptos in Greek means thin. Tin means thread, so this is thin threaded stage. Here chromatin fibers start condensation process, and they gives the appearance of chromosome or rod-like structure. Nucleus is distinct in this stage, and the chromosomes or chromatin fibers are intermingled with each other. They gives the appearance of a bouquet. So this stage is also known as bouquet stage. Next comes zygotin stage. Zygos means double, tin means thread, so this is known as double threaded stage. Here the formation of bivalent occur. That means a pair of homologous chromosome occurs in association with each other with the help of a structure which is known as synaptonymal complex, or synaptonymal complex यहाँ पे बनना शुरू होता है, and this process of pairing is known as synapsis. So synapsis begins in this stage. Next comes paketin stage. Pacus in Greek means thick, tin means thread, so this is known as thin threaded stage. In this stage, one of the most important event occurs, that is known as crossing over. So crossing over occurs in this stage of prophase 1. It involves exchange of segments of chromosome between two non-sister chromatids, which brings variation. That plays an important role in evolution. Next comes the diplotin stage. In this stage, there is formation of chiasma. It means there is separation of these homologous chromosomes by dissolution or breakdown of the synaptonymal complex, but these remains attached with each other at the point of crossing over, and that point is known as chiasma. Next comes diakinesis. Here, one important event occurs, that is known as terminalization. That means complete dissolution of this synaptonymal complex. So these are the various substages of prophase. So first stage, that is leptotin. In Greek means thin. Tin means thread, thin threaded stage. First stage, a nucleus In this first stage, nucleus size increases considerably. Here, the cell is metabolically active and it increases in its size. It starts with condensation of chromosome. जो chromosomes हैं वो condense होना शुरू होते हैं इस stage में by the process of coiling and dehydration. Then thread-like structures bearing beads like structures are called known as chromomeres. In this stage, chromatin fibers bear these bead-like structures, and these bead-like structures are known as chromomeres. In this stage, nucleolus is distinct in the nucleus. And this is the photograph which shows this leptotin stage. Here there is prominent nucleolus also, which is present in the nucleus. And chromosomes in this stage inside the nucleus appears like a bouquet. So they gives the appearance of a bouquet. So that's why this stage is also known as bouquet stage. इसलिए इस stage को bouquet stage भी कहा जाता है. Next comes zygotin. Zygos in Greek means double and tin means thread. So this is double or double threaded stage. Here the pairing of homologous chromosomes starts. So it begins when homologous chromosomes start pairing, and this is known as zygonema. In each pair of chromosome, one is derived from male parent and other from female parent. The pairing of chromosome throughout the entire length is known as synapsis. And this process of pairing is known as synapsis or conjugation. And pair of homologous chromosome is also known as bivalent. जो pair है homologous chromosome का उसे bivalent भी कहा जाता है. And there are three types of synapses. Basically जो synapsis formation है वो तीन तरह की होती है. First is pre-centric synapses. It means the pairing starts from the center or centromere. जब pairing start होती है centromere से, that type of synapsis is known as pre-centric. And then comes pro-terminal synapses. In this type of synapses, pairing of homologous chromosomes starts from the end and proceeds toward the centromere. जब जो pairing है वो start होती है end से and it ends at the center. So यहाँ से start होगा और it will end in the centromere region. Then third one is the random synapses. As the name indicate, it occurs at several points. ये several points पे start होती है and synapses continued along the length of chromosome until it is completed. और तब तक चलता है जब तक ये synapses का process complete नहीं हो जाता. So there are basically three types of synapses formation. That is precentric, proterminal, and random synapses. So this zygotin stage, it involves mainly the formation of bivalents. And these bivalents, it consists of 4 arms. These are also known as tetrad or termed as tetrad, while the single chromosome contains 2 arms, which is known as dyad. So these are some terms used for these chromatids, number of chromatids, on the basis of the number of chromatids. The next stage is Packetin stage. Pacus in Greek means thick, tin means thread. So this is thin threaded stage. It means chromosomes continues the process of condensation. Here it starts when the pairing is complete. This phase starts when the pairing is complete and the synapse chromosome continues to become short and thick. Here chromosomes with 2 visible chromatids is known as dyad. So chromosomes की जो 2 visible chromatids हैं उन्हें कहा था dyad. Bivalent thus becomes tetrad, और जो bivalent means pair of homologous chromosome है, that is also known as tetrad, or quadrivalent with 4 homologous chromatids. It means it consists of 4 chromosomal arms. So that's why this is also known as tetrad or quadrivalent. At tetrad stage, interchange of fragments between paternal and maternal sets of chromosome takes place. This is a very important event which occurs in this process and in this phase, and this process is known as crossing over, or इसे कहा जाता है crossing over. Here, the exchange of this chromosomal fragment occurs between chromatids of one chromatid of paternal chromosome and other chromatid of maternal chromosome, that is non-sister chromatids, and this process is known as crossing over. This mutual exchange between non-sister chromatids result in recombination, so it brings variation. There is mixing of some characters due to exchange of genetic fragments or chromosomal fragments. Enzymes involved in this process are endonucleases and ligases. Endonuclease helps in breakdown of chromosome, and ligase helps in joining of these fragments. So these 2 enzymes are involved in this process. Sites of crossing over, where the crossing over starts, are known as recombination nodule. So the term used for the sites where the crossing over occurs is known as recombination nodules, and these recombination nodules found associated with the synaptonymal complex during prophase 1. और ये जो recombination nodules हैं, they are always associated with this complex which is known as synaptonymal complex that helps in formation of the synapses. So this packaging stage, it involves exchange of these fragments, chromosome fragments between non-sister chromatins. And this process is known as crossing over. It brings the recombination which occurs between the two strands of chromosome. And the point where the chromosomes have shown crossing over, and this point is known as chiasma. So this is the packaging stage under microscope. Next comes diplotin stage. Diplos means double, tin means thread, double-threaded stage. It is marked by dissolution of synaptonymal complex. So here, the breakdown of that ladder-like structure, which is known as synaptonymal complex, starts here. Homologous chromosome separates from each other except at the sides of crossover. जो homologous pair हैं वो separate हो जाते हैं एक दूसरे से, except the site of crossover, and that point is known as chiasmata. This X-shaped structure is known as chiasmata. जो ये X-shaped structure बनता है, this structure is known as chiasmata. Number of chiasmata determines the length and shape of the chromosome. Longer chromosomes have more chiasmata than the shorter ones. It is understood. The chromosomes appear like cross in case of single chiasmata. When there is a single crossing over, it gives the appearance of a cross. But when there are two crossovers, then it gives the appearance of O or ring-shaped structure. So if there are two chiasmata region remains on the chromosome until they are separated in the anaphase. So जो chiasmata के हैं वहाँ पे chromosomes attached रहते हैं until the start of anaphase. There is one important fact that in oogenesis of human developing oocyte, in the fetal stage, this formation of oocyte stops at this stage, that is diplotin stage, before birth. This is very important event. Fact that oogenesis ceases at this stage before birth in the human female fetus. So this is the diplotin stage under microscope. Next comes diakinesis. It is the final stage of prophase 1 and is marked by a process which is known as terminalization of chiasmata. Chiasmata are the points where the crossing over has been occurred. Two chromosomes remain together by their extreme terminals located at the chromosome ends. So जो chromosomes हैं वो pair में रहते हैं by the extreme terminals. Chromosomes fully condensed. In this stage, they're fully condensed, and meiotic spindle assembled to prepare the homologous chromosomes for separation in anaphase. So the preparation for the separation by the spindle fiber starts here. Nucleolus and nuclear membrane disappear at the end of diakinesis, as we have studied in case of meiosis. And in the late prophase, these events occur, that is disappearance of nucleolus and nuclear membrane. So this is the photograph which shows diakinesis, which involves the process that is terminalization, that is separation of. Next comes the metaphase 1. It starts with attachment of spindle fibers to the chromosomes and chromosome alignment at the equator. So chromosomes they lie in the equator or equatorial plane or metaphase plate. Here the centromere of each chromosome is directed towards the opposite pole. जो दोनों क्रोमोसोम है होमोलोगस पेयर क्रोमोसोम है, इसका जो सेंट्रोमियर है वो दोनों अपोजिट पोल्स की तरफ फ्लाई करता है. So after at the time of anaphase they will move towards the opposite pole. The tetrads scattered in the cytoplasm move to the equator of the spindle and align in two parallel metaphase plates that we can see in this animated photograph. So जो दो plates हैं metaphase plates बनती हैं, they are parallel to each other. So chromosomes का pair lie करते हैं thus. Center of the cell, that is equator or metaphase plate. Each homologous chromosome has two kinetochores, one for each of its two chromatids. So दो kinetochores present होते हैं एक chromosome में, one for each of its two chromatids. Next comes anaphase 1. In this phase, there is separation of the chromosomes. So it is marked by the separation of homologous chromosomes from each other due to the shortening of chromosomal fibers or microtubules. जो microtubules हैं और ये spindle fibers हैं, इनकी shortening से जो chromosomes हैं, they move towards opposite poles, and this separation stage is known as anaphase 1. Each chromosome with its 2 chromatins and undivided centromere migrates towards the opposite poles and in the poles of the cell and is known as dyad. So this chromosome with 2 arms is known as dyad. So ये full chromosome move करता है towards the opposite pole. So that's why in this phase there is reduction in chromosome number. So chromosome number इस phase में half हो जाएगा. The process of separation is called disjunction. इस process को बोलते हैं disjunction, while the aggregation of chromosome is known as congregation. Actual reduction in the chromosome number occurs during this phase. So जो actual reduction होता है chromosome number में, that occurs in this phase, that is anaphase 1. Next, telophase 1, that is the last stage of meiosis 1. This stage is marked by movement of haploid set of chromosomes at each pole. So chromosome number half हो जाएगा और जो haploid set of chromosome का, that will move towards the opposite pole. And nuclear envelope is formed around the chromosome. जो हैप्लॉइड सेट है क्रोमोसोम का, उसके चारों तरफ न्यूक्लियर एनवेलप बन जाएगा एंड बिकम्स अनकॉइल. जो क्रोमोसोम्स हैं दे विल अनकॉइल एंड आर कन्वर्टेड इनटू क्रोमेटिन फाइबर्स. न्यूक्लियस रीअपीयर्स. जो न्यूक्लियस है फिर से रीअपीयर हो जाएगा एंड रिजल्ट्स इन द फॉर्मेशन ऑफ 2 न्यूक्लियाई विच आर फॉर्म्ड एज अ रिजल्ट ऑफ meiosis 1. हेयर स्पिंडल एंड एस्टर डिसअपियर. जो स्पिंडल फाइबर्स हैं और एस्टर्स हैं डिसअपियर होते हैं एनिमल सेल्स में. आफ्टर कैरियोकाइनेसिस साइटोकाइनेसिस occurs, that is division of cytoplasm, and 2 haploid cells are formed. और इसके रिजल्ट से 2 haploid cells बनते हैं. Each daughter cell has received only 1 chromosome from each homologous pair. So जो एक homologous pair था उसमें से एक daughter cell ने एक ही chromosome receive किया है. और this movement of chromosome is independent, or we can say this results in independent assortment, which also brings the variations. Stage between the meiosis 1 and meiosis 2 is called interkinesis. So the term used for the stage which occurs between meiosis 1 and meiosis 2 is known as interkinesis or interphase 2, और इसे interphase 2 भी कहा जाता है. Next comes meiosis 2. So this is similar to meiosis. जैसा meiosis हमने पढ़ा था, it is just similar to that of. So this is equational division. It is also known as homotypic division. And there occur 4 stages, that is prophase 2, metaphase 2, anaphase 2, and telophase 2. Haploid cell divides mitotically and results in 4 haploid cell formation in second phase. So total meiosis के result से 4 cells बनते हैं and these all cells are haploid. Next comes prophase 2. This prophase 2 is very short duration and the chromosome do not undergo appreciable changes. इसमें कोई खास changes नहीं आते chromosome में. The 2 chromatids of each chromosome remains loose except at the centromere point. जो 2 chromatids हैं वो separate रहती हैं एक दूसरे से except at the centromere point and become thicker and shorter at late prophase, as we have studied in meiosis. Their centromere remains still undivided. इस स्टेज में जो सेंट्रोमियर्स है वो डिवाइड नहीं करेंगे एंड द माइक्रोट्यूब्यूल्स गेट्स अरेंज्ड इन द फॉर्म ऑफ स्पिंडल एट द राइट एंगल टू द स्पिंडल ऑफ द फर्स्ट meiosis. जो फर्स्ट meiosis है उसके राइट एंगल में जो स्पिंडल फाइबर्स हैं वो बनेंगे. द न्यूक्लियर मेम्ब्रेन एंड न्यूक्लियस अगेन डिसअपियर एट द एंड ऑफ दिस प्रोफेज 2. इसमें जो न्यूक्लियर मेम्ब्रेन्स हैं और न्यूक्लियस है फिर से डिसअपियर हो जाएगा. नेक्स्ट कम्स मेटाफेज 2. The chromosomes moves in the middle region of the cell and are finally arranged to the equator or metaphase plate newly formed in the of the newly formed spindle. So this we can see here. Initially, the centrioles were in this location, but in meiosis 2, their alignment is right angle to the first division. So in this case, centrioles are arranged in this region, and they helps in formation of spindle fibers. That is right angle to the first division. The chromatids of each chromosomes are joined at the kinetochores. जो chromatids हैं वो जुड़ी होती हैं kinetochore centromere के पास, which also contain disc-like structure जिसे kinetochore कहा जाता है. The chromosomal microtubule extending from opposite poles of the spindle as in meiosis. The next phase is anaphase 2. It is just similar to that of mitosis. Here, the chromosome breaks from the centromere and each arm of the chromosome will move towards the opposite pole due to stretching of these interzonal fibers. So the two chromatids of each chromosome separates and moves to the opposite poles of the spindle due to the shortening of chromosomal microtubules and the stretching of interzonal microtubules of the spindle. So here, contraction of these tubular fibers occur, जिसकी वजह से ये जो क्रोमोसोम की एक आर्म है वो मूव करती है टुवर्ड्स अपोजिट पोल एंड जो ये सेपरेशन होती है क्रोमोसोम की, इट इज़ ड्यू टू ब्रेक डाउन ऑफ द सेंट्रोमियर. सेंटर से जो सेंट्रोमियर ब्रेक डाउन हो जाता है और ईच आर्म दैट इज हाफ ऑफ द क्रोमोसोम विल मूव टुवर्ड्स द अपोजिट pole. Here they are known as chromosomes. When they will reach to the opposite pole, they have the content of a full-fledged chromosome. Each pole and has a haploid number of chromosomes and the haploid amount of DNA. And the amount is 1/4 of the DNA present in the original cell which entered in the meiosis. As we have earlier discussed that during interphase of this meiosis, the DNA is replicated twice while the separation occurs. DNA is replicated once while the separation occurs twice. So, जो original content होगा haploid chromosome का, that is present in this half of the arm. Next comes telophase 2. Here the chromosome at each pole decondense and the nuclear membranes form around each set of chromosomes. So यहाँ पे जो chromosomes हैं they will reach at the opposite poles और यहाँ पे the nuclear membrane फिर से reappear होगी and there is appearance of nucleolus again. The spindle and ester disappear. The spindle fibers हैं और esters हैं वो disappear हो जाएंगे and nucleolus is formed in each nucleus. So this is telophase, and as a result of telophase there is formation of 4 cells. It is the part of karyokinesis. Then it is followed by the nuclear cytoplasmic division, that is cytokinesis. So this cytokinesis divides the cell into 2, and as a result of meiosis total 4 cells are formed. Next, cytokinesis, it is just similar to that of cytokinesis 1 of meiosis 1. So the process of cytokinesis is identical to cytokinesis 1, resulting in the division of cytoplasm of each of the 4 daughter cells. So that's as a result of that 4 cells are formed as a result of meiosis. And this is the animal cell. In animal cell it is accompanied by the process which is known as cleavage furrow method, while in case of plant cell it is formed by cell plate formation method. Next comes significance of meiosis. So the first significance is maintenance of constant chromosome number. So it helps in maintaining constant chromosome numbers in an organism. Sexual reproduction involves the production of special sex cells called gametes, which fuses to produce new organism. So formation of these gametes is occur with the help of this reductional division, that is meiosis. Oogenesis, it results in the formation of haploid ovum. Out of 4 cells, 3 are polar bodies and only 1 functional egg is formed. In case of spermatogenesis, 4 functional spermatids are formed, which are haploid as compared to their parent germ cell, that is primary spermatocyte or primary oocyte, which are diploid cells, and these are germ cells which undergoes this division, that is meiotic division, which results in the formation of haploid gametes. Each gamete must contain half the number of chromosomes of the parent, and it is therefore essential that a reductional division occur at this stage of the life cycle. So that helps in maintenance of the total chromosome number. Next comes the recombination of genes. So crossing over, which occur in packaging stage of meiosis 1 or prophase 1. So crossing over between non-sister chromatids of homologous chromosome during prophase 1 of meiosis 1 results in genetic recombination and brings variation. So that crossing over is responsible for bringing the variations, and variation act as an important source of evolution. Individual produced as a result of meiosis permits natural selection of the best selected species. So it impart important characters which helps in survival, and it plays very important role in evolution. Next, genetic variations. So due to independent assortment during meiosis, that also leads to some variations. So the random distribution and consequent independent assortment of the chromosomes during metaphase 1, at the time of anaphase 1 of meiosis 1, of meiosis produces new genetic combination. So that results in formation new genetic combination. As we can see, possibility 1 here, these chromosomes are marked by two different colors. One is blue color, other is the red color. So either these blue chromosomes can move to one cell, and reddish or red will move to other cells. So in other case, there can be mixing. So one chromosome is a blue color, other is a red color. So this is known as independent assortment. So these are the various combinations which can be obtained by these cells on the basis of their chromosomal distribution. So this chromosomal distribution helps in bringing the genetic variation in an organism. Next, repair of genetic defects. So the recombination that occurs in meiosis helps in the repair of genetic defects in next generation. Some genetic defects are repaired by this recombination. If a genetic defect occurs on a certain allele of one parent, that can be replaced. That diseased allele can be replaced with the healthy allele of the other parent, and thus allows the healthy offspring. So there are chances that can repair the genetic defects during the crossing over. So this was all about this meiosis. I hope people get some idea from this presentation. If you have any questions, queries, and any suggestions, you can give it in the comment section. Thanks for watching. Have a great day.